Work it, make it, do it, makes us harder, better, faster, stronger.
Until now, we have looked at a number of key characteristics of life. We have looked at cell structure. We have looked at how energy is metabolized. We have looked at some other characteristics of life. Now what we want to do is we want to go towards the concept of how does an organism reproduce itself. Now when you think about it, an organism to reproduce itself, or even a cell dividing, that information has to be transferred from one cell to another cell. Otherwise, you'd have cell A making cell B or cell C. But cell A has to make another cell that looks reasonably like itself. So how is this information passed on from one cell to another cell, from one organism to another organism? So basically, we're setting ourselves up for reproduction. But getting back to the main point as to how information is transferred from one cell to another cell. The way this is done is through a molecule called DNA. DNA is the blueprint of life. Now, prior to the 1950s, a number of scientists had postulated as to the nature of information transfer. Initially, it was thought to be proteins. However, as more information came into place, it was reasoned that a molecule called DNA was carrying the information that is transferred from one cell to another cell during the process of reproduction or cell division. Now, the discoverers of this molecule, a macromolecule called DNA, were Watson and Crick. They, with other scientists, organized, researched, and figured out exactly how this process works. Now, today, we're quite certain that DNA is the blueprint of all life. Now, let's look at the DNA molecule. First of all, the DNA molecule is a double-stranded molecule made up of nucleotides. If you would look at a nucleotide is made up of a phosphate group, a sugar, and a base. Now, DNA will have 4 different types of nucleotides. Each nucleotide will have a different base. For example, you have adenine, guanine, cytosine, and thymine. Each of these bases will combine with a sugar and a phosphate to form a nucleotide. As you can see the double-stranded molecule to your right, you can see that the molecule looks like a ladder. The sides of the ladder are made up of sugar-phosphate backbone, and the steps of the ladder are made up of the bases that combine with each other. If you look carefully, you'll see that the adenine combines with a thymine, and guanine will combine with a cytosine through depicted hydrogen bonding. Now that's important because what we're saying is that a molecule of DNA is made up of 2 strands, and the strands are held together very specifically by complementary bases. Therefore, an adenine will combine with a thymine, and a guanine will combine with a cytosine. Now, what determines the genetic code which is embedded in this molecule? The genetic code is determined by the sequence of nucleotides that run up the sides of the ladder or the DNA strands. For example, I'm looking at this molecule here, and I see my sequences A, T, G, and A. Now my complementary sequence, which is the opposite, is going to be T, A, C, and T. As we started off, the purpose of trying to understand DNA is to appreciate how information is passed from one organism to another organism. When an organism goes through the process of replication, the DNA has to double. Now, this process is called DNA replication. In this slide, we see in a very simplified illustration how DNA doubles itself. What happens, as you can see, the double-stranded DNA consists of two strands. One is actually complementary to the other, almost the opposite of the other. An A will combine with a T, a G will combine with a C. So as you look at this double-stranded molecule, you see it starts to unwind. When it unwinds, the double strand unwinds, each strand will now be copied to form a complementary strand. So an old strand will now come in and form another strand that is the opposite of it. And as you can see, what the bottom line is, one molecule will form two molecules. We will go much more into the process of DNA replication later on in this course. But for now, understand that DNA replication is a very simple process for DNA basically doubling, and as it doubles, the information is transferred from one molecule to two new molecules. Now, when we look at DNA replication again, we're looking at it in a little more detail here. Again, each strand is copied to form a complementary strand. As you can see, the nucleotide, look at the nucleotide sequence. The sequence on the left, as you can see from the top, is GTAGCT. To the right, it's CATCGA. My molecule has unwound. Nucleotides then come in and will attach themselves in the process of DNA replication so that wherever there is a G, a C will come in, wherever there's a T, an A will come in, and so on. As this process goes, you can see how the nucleotides from the double-stranded molecule have now formed two new double-stranded molecules. Now, this process is under the control of a complicated series of enzymes that specifically know where to attach a given nucleotide. Now, in the process of DNA replication, this process has to work extremely well, otherwise you have the wrong sequence of nucleotides incorporated. When this happens, we have what we may call a mutation. Now, mutations take place on a continuous basis. However, an organism knows how internally to repair problems or mistakes that occur. Let's look at our molecule on the left that we will replicate. Now, as you can see, we start with a double-stranded molecule. On the left, the sequence is GTCGCT. When that molecule replicates itself through the process of DNA replication, you see suddenly where the arrow points, the G gets converted to an A. Well, that means that the new sequence is going to be GTCACT, which means that we have now a point mutation, that the process of DNA replication has produced a new sequence of DNA, which means that this new sequence is mutated, and therefore that particular organism may carry on new characteristics. So let's review some key points on DNA. First of all, DNA contains the genetic code. Secondly, DNA is present in all living organisms, and we will find later on, for the most part, it is present inside the nucleus of a cell. Number 3, the basic genetic code is the same for all organisms on Earth. Now, genetic makeup will differ from species, but the overall genetic code is the same for a human as it is for a fly. Lastly, genes in an organism may range from a few to many thousands. For example, a simple virus may have perhaps 5 genes, whereas a human will have upwards of 30,000 genes. The more complex an organism is, as you might expect, the more genes it will have.